Hello everyone, this is Dr. Suresh again, and in this video I'll be talking about gluconeogenesis. So in our previous videos we have studied about the degradative pathways of glucose, that means glycolysis and the Krebs cycle, and here gluconeogenesis. When you don't have glucose, then how the glycolysis and TCA cycles will be taking place. Okay, for these glycolysis and TCA cycles to take place, there is a absolute need of glucose. But what to do when your body doesn't have the adequate levels of glucose? So for that we have another alternative pathways in our body to produce glucose. So one among such, that is gluconeogenesis. Gluconeogenesis is nothing but synthesis of glucose from non-carbohydrate sources or precursors. Okay, so the synthesis of glucose from carbohydrate source, that is nothing but glycogen. Okay, so glycogen will be completely broken down in case of starvation. Okay, and it will be utilized. Then what else? What happens if there is no glycogen storage? Okay, so that's what the definition of gluconeogenesis is: synthesis of glucose from non-carbohydrate precursors. We'll see where, as I said, the location of pathway is very important. So glycolysis is in cytoplasm, TCA cycle is in mitochondria. Again, gluconeogenesis, the tissue where it will be taking place is liver. So remember, not like other pathways, gluconeogenesis specifically takes place in liver. Liver is the sole site for production of glucose. Okay, though different substrates will be coming from the different parts of the body, but liver is the sole site for production of glucose. Okay, and specifically this takes place in case of starvation, and kidney also capable of making glucose little amount by gluconeogenesis next to the liver. And this gluconeogenesis will take place when there is a decreased levels of glucose in circulation or in the body. So which conditions? Like starvation. Otherwise it is also known as precursors, like gluconeogenesis precursors. Okay, lactate, glycerol, and where you come across this lactate, glycerol, like what to say, lactate, what is the end product of glycolysis? That is pyruvate. So in absence of oxygen, this pyruvate can be converted to lactate, right? So here non-carbohydrate source will be lactate or pyruvate. Pyruvate also you can include. Okay, and glycerol, when you broken down the tricyl glycerols, so TAG is nothing but, okay, glycerol plus 3 fatty acids, and these 3 fatty acids undergo beta oxidation to form acetyl CoA, and they enter into TCA cycle. And what is the fate of this glycerol? This glycerol enter into gluconeogenesis, and it will be shifted to liver, and there it will entertain to make glucose. And glucogenic amino acid, as I said, when you broke down the protein, okay, it gives rise to amino acids, and amino acids such as alanine. Okay, the difference, pyruvate is a 3-carbon compound, alanine is a 3-carbon compound. Pyruvate is a keto acid, alanine is amino acid. So when you remove the amino group, alanine will be converted to pyruvate. Alanine will be converted to pyruvate, and this pyruvate again converted to some other substance and enter into gluconeogenesis. Propionic acid or chain fatty acid broke down, where you will get propionic acid, and intermediates of TCA cycles such as oxaloacetate, okay, alpha ketoglutarate via glutamate formation, they will be involving fumarate like this. They will be involving in making of glucose. So the proper definition of gluconeogenesis is synthesis of glucose from non-carbohydrate sources such as pyruvate, lactate, glycerol, because ultimately when pyruvate is available, also pyruvate has to convert into lactate. Okay, and glycerol from fatty acid broke down, I mean TAG degradation, gluconeogenic amino acids such as alanine, aspartate, and then glutamic acid, propionic acid coming from the out chain fatty acids breakdown, and intermediates of TCA cycle such as oxaloacetate, fumarate. Okay, they're all involved in gluconeogenesis. Okay, so you see here the picture. When you see the picture here, citric acid cycle is a common substrate. If you take as glucose, I mean the reversal, okay, you can make out. So lactate can be converted into pyruvate, and pyruvate can be entered into one of the intermediates of TCA cycle, and finally is forming phosphoenol pyruvate, and phosphoenol pyruvate to glycerolide, and then glucose. Same way, glucogenic amino acid will be entering into one of the intermediates of TCA cycle. Propionic acid is also one of the intermediates of TCA cycle. Glycerol is solely converted into glycerolide 3-phosphate and converted to glucose. So this is the overall mechanism of how non-carbohydrate sources entries into the pathway of gluconeogenesis. Right, first we will see one by one. Conversion of propionate to succinyl-CoA. So this propionate will be converted to propionyl-CoA by the enzyme acyl-CoA synthetase, okay, where ATP is converted into AMP plus PPI. Here remember, you might have got doubt, ATP has to form ADP, then how come AMP plus PPI? Here two phosphate groups will be removed from ATP, okay, to form AMP, okay. Here phosphate will not be utilized, but the energy which is releasing, okay, removal of one phosphate from ATP will give you 7.2 kilocalories. Okay, similarly, one more phosphate removal will give you another 7.2 kilocalories, which total of 14.4 kilocalories, which will be involved in making of propionyl-CoA. And this propionyl-CoA converted to methyl malonyl-CoA, okay, by the enzyme propionyl-CoA carboxylase. Propionyl-CoA carboxylase, one more carbon is added, carboxylation reaction. This is carboxylation reaction where again there is a need of ATP with the help of water, D-methynyl malonyl-CoA, and this D-methynyl malonyl-CoA converted to L-methynyl-CoA. So there is a form, that means hydroxyl group, OH group present on the next to penultimate carbon on the right side or left side. So in D-methynyl malonyl-CoA it is on the right side, and no L-methyl malonyl-CoA it is on the left side. So and this L-methyl malonyl-CoA converted to succinyl-CoA, one of the intermediates of TCA cycle. Okay, so here to convert the methyl malonyl-CoA to succinyl-CoA require vitamin B12. So how? The removed methyl group will be taken by B12 to form methyl cobalamin. Here methyl malonyl-CoA is acting as a source of methyl group. Okay, so with the help of B12 only you can form succinyl-CoA TCA cycle. So in case of B12 deficiency, this reaction will not take place. So you will be deficient of one of the source in making of glucose from propionic acid. Next is conversion of glycerol to dihydroxyacetone phosphate. Okay, so how glycerol? So as I mentioned, okay, this is glycerol. Okay, 3 fatty acids. This is first fatty acid, second and third. Okay, so when this is TAG, that means 3 fatty acids attached to one glycerol molecule. So when there is a breakdown of TAG, so it forms glycerol and 3 fatty acids, and these 3 fatty acids will undergo beta oxidation, okay, and forms acetyl-CoA, and TCA cycle gives energy. Okay, but here this glycerol in the circulation or from the adipose tissue will straight away enter into the circulation and reaches to the liver. Okay, in liver it will be converted to glycerol 3-phosphate. Okay, the donor of phosphate group here is ATP. ATP is converted to ADP, okay, by the enzyme glycerol kinase. Remember, glycerol kinase specifically present in liver cells, in no other cell. Okay, glycerol kinase specifically present in liver cells, in no other cells. And glycerol 3-phosphate again with the help of accepting 2 hydrogens or removing 2 hydrogens. Okay, so the interconversion, okay, this glycerol is alcohol form is converted to keto form, okay, by the enzyme glycerol 3-phosphate dehydrogenase. So this converted to dihydroxyacetone phosphate. Okay, so where NAD can be converted to NADH2 by removing 2 hydrogens, okay, which is reversible, this one. And this dihydroxyacetone phosphate again converted to glyceroldehyde 3-phosphate, again glyceroldehyde 3-phosphate, 2 molecules combine to form fructose 1,6-bisphosphate. So again to make glucose finally. Okay, so remember, if you 2-3 steps, it is a totally reversal of glycolysis only. So what we can think of, someone will say, is gluconeogenesis is a reversal of, complete reversal of glycolysis? No, because remember there are 3 irreversible steps in the glycolysis. Okay, so otherwise what happened? Glucose to pyruvate and pyruvate to glucose, it is easily reversible, but 3 steps making irreversible. So you have to say gluconeogenesis is not a reversible of glycolysis. Okay, so in order to make it reversible, you have to bypass the 3 reaction, that is the 10th step, 3rd step and 1st step. Okay, you need separate set of enzymes which you can making is possible. So the next thing is how you are getting one of the substrate in gluconeogenesis, that is lactic acid. Okay, so lactic acid is mainly produced in the skeletal muscles. Okay, so as I said, not similar to liver, skeletal muscles, there is a continuous happening of glycolysis. Okay, because there is a contraction of muscles in continuous process, so they need continuous energy. So glycolysis need not to stop. So for that, what happened? The glucose, some of the glucose will be again generating lactate, again it will be transported to liver to again it will be transported to glucose. There is no production of glucose in the muscle, skeletal muscle. So here, this process of synthesis of glucose from lactate, otherwise known as Cori cycle, how it is circulated. You see here in skeletal muscle, glucose undergoing continuous glycolysis produces pyruvate to lactate, and this lactate will be transported to the liver. So that is the reason behind when you hit the gym all of a sudden and done with the strenuous exercise, next day you will be ended up with the soreness of the body and body pains because of the accumulation of lactate. Okay, because of the accumulation of lactate in the muscles, you will be feeling pain or and this body pain will be subsided within the 2-3 days as the accumulated lactate will be washed off to liver. Okay, and this lactate will be transported here. As I said, a formation of lactate from pyruvate is a reversible reaction. So pyruvate can be formed from lactate can be formed from pyruvate. So once lactate reaches to the liver, it can convert it back to pyruvate, and pyruvate will be converted to oxaloacetate, and oxaloacetate again converted back to phosphoenol pyruvate, because directly the formation of pyruvate to phosphoenol pyruvate is not possible because it is a reversible step. So to bypass it, so you have to convert pyruvate into another compound, oxaloacetate, and this oxaloacetate again converted to phosphoenol pyruvate by carboxylation and decarboxylation reaction, and the synthesized glucose will be transported again back to the skeletal muscle for continuous glycolysis for producing energy. So this is the overall mechanism of Cori cycle. So glucose-alanine cycle. So this is from the synthesis of glucose from non-essential amino acids such as proteins, you can say. So you see what is happening here. So as you don't have glucose, okay, so the source of glucose will be here, non-essential amino acids. Glucose will be converted to pyruvate, and I told you the difference between pyruvate and alanine is the amino group. When you add amino group to pyruvate via transamination reaction, it will be converted to alanine, and this alanine will be transported back to liver, where again it will be converted to pyruvate, and this pyruvate will be converted to phosphoenol pyruvate via another formation of oxaloacetate, and it will form glucose and produce glucose again back to the circulation, again to the skeletal muscle. This is a cycle. This is known as glucose-alanine cycle or Cahill cycle. Okay, so these are all the things you can make out how glucose is forming, like from non-carbohydrate sources.